So this dude named Calvin, he did this cool experiment, which is called the lollipop experiment, which we'll talk about in a second, to show that how carbon is added to ribulose bisphosphate. So that's basically what we're going to show here. And there's two scientific techniques that allowed him to do this. But let's just look at the setup here real quick. So here, this looks kind of like a green lollipop, right? In this vessel was filled with algae. Algae do photosynthesis. They take the light's energy and make carbon compounds. So what he did was he just gave it light. I'm not sure why my voice is so high. He gave it normal carbon dioxide. He also gave it another source of carbon dioxide, hydrogen carbonate, and he just let them do their normal thing. And they were making all the carbon compounds. So we talked about how photosynthesis produces glucose, but also we make other carbon compounds in plants. I say we, they. So, it also needs amino acids. That's made of carbon, right? It also makes lipids for its lipid membrane. It's going to generate nucleotides for its DNA and RNA. So it does. It makes all of these things, which can be made from that original glucose phosphate. So we have to remember that as well. So then, after being exposed to normal carbons, he gave it radioactive carbon. So radioactive carbon is also known as carbon-14. And now this carbon-14, now that we know how the light-independent reaction works, this carbon-14, if it's attached with CO2, will bind, right, to that ribulose bisphosphate, and we can see it show up. So there's two techniques that allowed him to see how this would show up. So he separated the different products, amino acids, lipids, and nucleotides, also some sugars. He separated the different products using chromatography. The other thing, which is kind of hard to see here, let me just move this up. The other technique he used was autoradiography. Autoradiography is this technique of taking a picture and seeing what's radioactive to observe. That's not how you spell observe. That's okay. Radioactivity. So it's almost like taking an X-ray to see like what's radioactive. So that was his technique. So when he did his chromatography, we can look here. This is a sample of his chromatography here. He did two different things. So one, he did a chromatography immediately after he introduced that radioactive carbon dioxide. You can see here 5 seconds. And you're looking at all these molecules. Alanine is amino acid, malic acid. Here's our triose phosphate, if you remember, that was made right before glucose. There's a sugar down here. And then right here, PGA. So PGA, I want you to think of PGA and glycerate 3-phosphate as kind of the same thing. These things happen immediately when that carbon dioxide is bound to that ribulose bisphosphate. So these are our first molecules that are made. And that's what Calvin was able to show. He was able to show that CO2, the very first molecule it makes, is this glycerate 3-phosphate, which is an example of a PGA here. And then down here, just to further elucidate some other things it makes, 30 seconds after photosynthesis, only 30 seconds, which I think is pretty amazing, all of a sudden you see all of these other radioactive molecules. So alanine is being made already. Serine is radioactive here. It's a darker stain. Aspartic acid, another amino acid. Our triose phosphate, right? That's now radioactive. Some other disaccharides down here. That was his technique that he used. He used both autoradiography and chromatography to show that the very first carbon molecule that we make is glycerate 3-phosphate.